The human serotonin transporter, also known as the 5-HT transporter or solute carrier family 6, member 4, is a sodium-dependent serotonin transporter. It is classified as a monoamine transporter protein and is found in Homo sapiens. It belongs to the neurotransmitter symporter family and is encoded by the SLC64 gene. SERT acts in transporting serotonin across from the synaptic cleft back into the presynaptic terminal. Termination of serotonin signaling occurs as sodium and chloride are recycled back into the presynaptic neuron. This process is depicted by the catalytic activity formula below. SLC64 has two alleles known as long and short. These change the travelling times of the signals and are closely related to both psychological and psychopathic disorders in humans. In regards to the biology, the action potential initiates the opening of voltage-gated ion channels in the presynaptic terminal. The ion influx results in 5-HT vesicles fusing to the plasma membrane and then release into the synaptic cleft. The serotonin transporter is responsible for the reuptake of serotonin, also known as 5-HT, into the presynaptic neurons. This is powered by a symport mechanism using concentration gradients. Upon reuptake, 5-HT is metabolized into an inactive metabolite by monoamine oxidase. To summarize the mechanism of the transporter, two sodium ion binding sites are exposed to the extracellular environment. Upon binding to sodium ions, these sites transition from an inwards to an outwards facing conformation. 5-HT substrate and sodium ions are released into the presynaptic terminal, and the protein will return to its outward facing state. S-citalopram, a serotonin reuptake inhibitor, binds to the transporter's orthosteric active site and inhibits 5-HT reuptake abilities in a process that locks SERT into an outwards open conformation. This blocks serotonin from binding to key locations, increasing the 5-HT in the synaptic cleft. Bioinformatics data displayed here depicts the respective molecular weights and isoelectric points for the asymmetric and biological units. For the sequence derived from UniProt, the total number of positively charged residues is 40, and the number of negatively charged residues is 45. The isoelectric point for the UniProt sequence is 5.89 with a molecular mass of approximately 70,000 kilodaltons, while for the PDP sequence, the isoelectric point is 8.31 and the mass is approximately 62,000 kilodaltons. Here is the relevant primary structure. The UniProt accession number is indicated at the top. Many of the essential amino acids in the process of 5-HT transport and blocking of reuptake by S-citalopram are emphasized. For crystallization, thrombage cleavage sites were inserted into the N and C terminuses at the residues glutamine 76 and threonine 618. To increase thermostability, various mutations were introduced. Tyrosine 110 and isoleucine 291 were mutated to alanine, and threonine 439 to serine. Additionally, the surface-exposed cysteine residues were also mutated to alanine, that being cysteine 554 and cysteine 580. Important residues for the sodium 1 and 2 binding sites are emphasized in dark green. There is also a disulfide bridge at the cysteines 200 to 209. The secondary structure of SERT consists of numerous alpha helices and beta sheets. SERT has three domains: the transmembrane domain, the extracellular domain, and the intracellular domain. There are 12 transmembrane domains in total, and each is stabilized by H-bonds. For example, glutamine 136, which connects nitrogen to hydrogen groups on the 6 transmembrane backbone. The extracellular and intracellular loops of SERT contain several secondary structures such as beta strands, beta turns, and alpha helix hairpins. These are stabilized by hydrogen bonds between the backbone atoms of the polypeptide chain. The SERT protein is arranged in a specified spatial arrangement to allow for the tertiary structure and function of the protein, and a globular shape. The 12 transmembrane domains are arranged in a way that creates a binding site for serotonin and other ligands through the establishment of a hydrophobic channel. The extracellular and intracellular loops of SERT are important for substrate binding and transport, and can regulate SERT activity. These structures are established by a combination of H-bonds, electrostatic interactions, van der Waals forces, and hydrophobic interactions. Secondary structures on the protein are again emphasized here. The alpha helix is in blue, with the number of beta-pleated sheets of the additional chains in red. The transmembrane helices are arranged in a bundle-like structure, with loops connecting the helices. The loops are important for binding and transport of serotonin, as well as regulation of SERT functions. The active site of SERT is located within the central cavity over here, which is performed by the arrangement of transmembrane helices and is composed of several amino acid residues that are essential in the binding and transport of serotonin. Key residues in the active site of SERT include aspartic acid residue 98, and aspartic acid residue 437, highlighted in yellow. Additionally, serine 439 is highlighted in blue. These residues are involved in the binding of 5-HT and the binding and dissociation of sodium ions. These help establish the concentration gradient required for transport across the membrane. Phenylalanine 335 plays a role in substrate recognition and selectivity, highlighted in orange. If we zoom into the active side a bit more, the mechanism for serotonin binding and reuptake by inhibiting drugs can be summarized as following. Upon the initiation of binding, isoleucine 108 forms hydrophobic contacts with the indole ring of 5-HT. From here, 5-HT will diffuse inwards in the direction of the allosteric site and become stabilized by interactions with phenylalanine 335 and arginine 104. The binding to the orthosteric region occurs as the indole group of 5-HT forms aromatic interactions with tyrosine 95 and phenylalanine 341. And the 5-HT amine group disrupts the hydrogen bond between aspartic acid 98 and tyrosine 176. The 5-HT aromatic ring then positions between tyrosine 95 and valine 343. The amine groups of S-citalopram also interact with aspartic acid 98. Tyrosine 176 experiences interactions with S-citalopram's hydrophobic ring structure groups. Isoleucine 172 is also indicated to be important for 5-HT transport and is greatly affected by S-citalopram. Additionally, phenylalanine 335 forms edge-to-face aromatic interactions with S-citalopram, which acts to prop transmembrane 6 into an outward open conformation. In this image, S-citalopram is centered within the hydrophobic channel established by the transmembrane domains. At this location, the drug is able to inhibit the reuptake of 5-HT back into the presynaptic terminal.